So the new form of energies are being made by the atomination structures of helicals. They make a structural analysis for transferring the cycles of circadian states of the controlling of polys and all the structures from VE to BA and the configurations of NS shells of NS3 and P4 or the different configs of from cis to the mediate protein. The configs are CM plus 2X. They form a centripetal network for transferring the electrovalency of oxidative positions. Yes. So the energies have their non-ability of d orbitals for bonding. They are the anomalous behavior of some atomination structures. The tendency to form multiple bonds, the first member of each group differs from the heavier members. So this ability to form p pi bonds, multiple bonds, help to configure the second row elements. So the configuration of inert pair groups helps to transfer the families of production energies. So next we move on here is the biomolecular energy has been changed into a new form of cysteine proliferation. The proliferative activities are in the vitro proliferations. We have talked about the energies which are being made by the configurations. So cysteine is a very cis complexity phases which make a prophase or a telomere phase for the formations of proliferative activities in Drosophila. They make uh, gamma structural proteins for configuration of it only. So the agile mammillus is new form of vitro proliferation, and also the electron proteonomic cisplatin. So agile mammillus is the medicine biomedical medicine which controls the cisplatin, chromomycin, cytosine, arabinoid, and pyrrolysine, and also the antidiabetic activities. They make also inhibit the vitro proliferation of human tumor cell lines. So they have a proteonomical thing for configuration of it. So oxidative reaction with ephedra and conscious reactions of biocentrism in the bodily structure and the parabolical activities. In the oxidative positions, the ephedra is a non-adrenaline thing which keeps on changing the reaction cycles of cyclic helicals which make a family of proteins. In this way, the formation of cycles through the cis or amylations, the energies are being transferred, the changes in their own. So the parabolical activities are being done in the biocentric manner. So the biocentric is the manner which can be controlled by the synaptosomes. The synaptosomes activities, one molecular energy controls all the molecular energy because of its genetical coding. Because of its genetical coding, the energy have its own shell membranes, which have their energy to attract or or make a whisker on a network of series of reactions. So the energies are being made by it. Energy of molecular changes in some kinases, pairs, chemical equilibrium in stoichiometry, redesigning the anatomy. So stoichiometry, the chemical equilibrium is there in the stoichiometry. The chemicals make configurations in it and give their pairs to another biological thing to make a chemical equivalent for thermodynamical changes. So the in stoichiometry, the energies are being metabolized in the things of generating the anti-aging chemicals. They make configurations for balancing the structures of proteins, or they can make the glyconeogenesis or C14 or by the hydroxide tryptamines. So that tryptamines are the tryptophans which all make the configurations in the amines group for the formation of new energies for the cellular fluidic pathways, and these are the main things which configure them for their states of redesigning the matter. So the next is the stereomyocentromere. Pericentric cycle is movement in synapses and formation of mitotic activities in Drosophila. So the Drosophila is a nematoda species which have a metabolic system for controlling the configuration of unwinding their DNA or controlling the mRNA positions for their configurations of proteins. By doing it, they can make a hibernated network series which can be on or off at that moment. In this way they can make a renewing amination. So the chemicals configure themselves by the formation of mitotic activities by cis complexity tryptophan and the energies which configure it. P2 dicentric apopopulous is the main apolis which concentrate their bicentric energies. So the energies make the configuration of chemicals which have their own um, properties of alkynes and electrovalencies of ns pair and plus 4 pairs. So the photochemicals like anacardium which controls the activities of the brain by activating the memory factor. Memory is the precortical index for transferring the data structures from the apolytic cell formation. Uh, the proteins are being made by the order of the brain. They also make a Thujon barrier for their configs. So the next topic we will move here is so here is the new configs that we have talked about. The progenitors in the stem cell have a stereogenic mitotic in germline protein. The folate proteins are used to metabolize the things for configurations of heme or fabricated protein. So they activate the PZ for their polymorphic activities and also the photoconverter to make hydroxyl groups of GLP-1 FORL1. So the folates change their atomization structures by activating some of the cells. The mitotic activities are the main example of it. The mitotic process, which configure the energy cells, the sperma, metheca, and the oocytes, and the embryos which are being made by the it. So the in every organism there are different types of spermatheca. They make a family of their own inert pair. Like they can make a triple X or triple Y for because they have a genetical dark structure for transferring it. So here is the structure which I am talking about, the levels of photoconverter in dendra to S2B. You can see it that the configurations are there by it. So the second is the so the second we will talk about here is the configurations of RKLA protein for metastates. So RKLA is a type of ryanodic abenose probe to configure the pathways of cellular proliferation and signaling intercellular coding. So the STF energies for beta structures for beta metabolism is there, which overcomes that action. CEWNK1 is the nematode which have a dogma gamma and unwind the DNA which amidate the perform their nitrohedra function configuring the helical rack in C. elegans. They perform their isopathic functions in their replicated tRNA. So they make a isopath for being passed through a structural analysis of alpha units. They are being passed from it. Because of it, the physical age is also changes from the real age. Because of the some mutated chemicals are being make their own chemical configuration for passing it. The electrovalency they make to pass all the energies for formation of things, the main topic of it. uh, so the function they replicate is the tRNA. Here we can see it, the structures which make it, which are the highest structure and types of strands cycle of chemicals. So HTF energies are the hydroxylating tetraboroacetate and the folate proteins which make all the energies. So the we know that the physical age is also changes and the chemical age is also changes because of the configurations of by the tumors and cell signaling. If you like the video, please like it and comment on this video. And also, if you not subscribe it, please subscribe it. Thank you for